I would like to thank the organizers for the invitation, but particularly for putting these nice presentations before mine, so I don't have to explain many of the concepts. As we all know, pregnancy at high altitude affects many millions of people around the world. And one of the major effects of high altitude and pregnancy is that the reduced fetal growth and the pregnancy rise in uterine artery blood flow. There is a particularly advantage of doing studies in Bolivia. One of them is like that there is a big population living above 2,500 meters. And even like big part of this population lives even higher than 3,500 meters. And I would like to talk about a little bit the background of this work that we have been doing in the past. And it started with studies led by Lorna and Colleen, where they found that Andeans are protected from altitude-associated reduction in fetal growth. Also from Dino's lab as well. So one of the papers, for instance, this one from Dino's lab, and that Colleen showed before, it's a birth weight curve showing that in Andean populations have a shift in the curve compared with European, showing here in blue at high altitude. And that is not reflected at low altitudes. Also, there is, as Colleen showed previously, there is an increase in blood flow, in uterine artery blood flow. And this is shown in 20 and 36 weeks. In the Andean populations, there is an increase, whereas in the European, this rise is not as pronounced. Again, Colleen already showed this, but I just wanted to point out that genetic studies show different candidate genes. One of them, PRKA, which encodes for the MPK protein. And again, this is showing just the relationship between birth weight and the different variants of MPK. And you can see here that at high altitude in the open symbols, the Andean variant is increases the birth weight. So MPK is, as it was told before, is a metabolic sensor. It has many inputs and many outputs as well. So activation by ischemia, exercise, starvation, by hypoxia as well. And it has catabolic and anabolic processes as the output. We are interested in the effect of hypoxia and how it can regulate MPK activity, and particularly how certain compounds can modify MPK activation. So we have access to different activators of MPK, also inhibitors and indirect activators as well. And how this leads to vasodilation. So here just to show you an example of, this is a pre-constricted aortic ring from mice. And it's showing that ACAR, which is an activator of MPK, induce a relaxation of these pre-constricted arteries, whereas in the knockout mouse, there is no effect of ACAR. So studies led by Dr. Moore show that in mice, using isolating the uterine artery and then using wire myography, they found that MPK activation by this A76 compound shifted the curve, the vasoconstrictor curve, to the right, and this shift is more pronounced in hypoxic conditions. So it seems like the functional effect of hypoxia will be to increase the vasodilatory responses to MPK activation. Further studies in our lab shown that mice exposed to hypoxic conditions during pregnancy, but also treated with ACAR, which is the other activator of AMPK, show that there is a partial prevention in fetal growth restriction induced by hypoxia. So in the control wild-type mice, control mice, in normoxia, there is no effect of ACAR, whereas in the hypoxia without ACAR, there is a reduction in fetal growth restriction, and that is prevented by the treatment with ACAR. And these were pellets implanted in the mice before the hypoxic condition exposure. So that led to some human studies that we developed in Colorado. So here's like the map of Colorado, and there's like a different altitudes, and you can see there is like a big area where the plains are, and this is where the Rocky Mountains are. And we study pregnant women that were delivering at Frisco. I'm showing a picture of that as a very nice little mountain town. And this hospital attends people living in the Summit County or the surrounding areas. So our cutoff was people living above 2,500 meters. We collected myometrial tissue from C-sections under two different conditions. One is appropriate for gestational age, and also IUGRs, and at two different altitudes. So we consider Denver as a low altitude site for pregnancy purposes. Probably an athlete will say that it's not low altitude because for exercise it's completely different. But in high altitude, as I say, above 2,500 meters. So we isolate these myometrial arteries that are embedded in the myometrial tissue, and they are the resistance size arteries. So they are responsible for controlling blood flow. We isolated these arteries, we mounted in a wire myograph, and we did some vascular studies. So the first study that we published out of this data showed that high altitude reduces the nitric oxide dependent vasodilatory responses. And I'm not going to show that because that's not the focus of this talk, but a following study we did, we tested A76 compound, which is the AMPK activator, in arteries that were pre-constricted to phenylephrine. And as you can see in the low altitude, in Denver altitude, we observed a vasodilatory response or a decrease in the preconstriction, that it was dose dependent. And in the high altitude tissue, we saw an increase in the sensitivity to vasodilatory response induced by AMPK. So we wanted to study also the mechanism for this. So these are the same graphs that I showed before, but just separated between low altitude and high altitude. And these are both appropriate for gestational age pregnancies. So when we use LNAME, which is an inhibitor of the endothelial nitric oxide synthase, to determine how much of the vasodilatory response was due to nitric oxide activation or production, we didn't see any effect of this. When we include another endothelial pathway inhibitor in the methacin, which blocks COX-2, so the production of prostaglandins, we didn't see any effect also in this response. When we mechanically remove the endothelium, we didn't see any change in the low altitude. So it seems like it's not dependent on endothelium signaling. However, in the high altitude, we saw a difference when we blocked nitric oxide synthase in the AMPK activation vasodilatory response. We didn't see a further increase when we block prostaglandin production, and we see like a mixed effect when we remove endothelium. When we compare also this with IUGR pregnancies, so in black is appropriate for gestational age, and in red is IUGR, We didn't see an effect on low altitude, but in high altitude, we see that there is although it's not significant, there is a trend to reduction in the AMPK-activated responses. So based on this data, our working model is, and as I said before, I didn't show all the vasodilatory responses, but we know that acetylcholine and bradykinin are also involved in these responses. So in the AGA, low altitude, all the vasodilatory responses are mediated by bradykinin and acetylcholine, are through endothelial nitric oxide synthase activation, and partially, a very small amount is due to AMPK, which we think that is mostly in the smooth muscle cells. However, in AGA high altitude, acetylcholine responses are reduced, bradykinin responses are maintained, and AMPK is increased. So AMPK, we think that is increased due to eNOS activation, but also partially by an eNOS-independent mechanism that we don't know. And there is still some AMPK activation in the smooth muscle cells that can be induced in relaxation. In the case of the IUGR, at any altitude, what we think is that the AMPK responses are impaired, as well as the cholinergic responses. Bradykinin seems to be maintained. So we think that the AMPK activation at high altitude might be a compensatory mechanism to maintain vasodilatory responses. So moving on, what is the effect of AMPK in the placenta? There are different studies, previous studies that show that either, for instance, in mice, show that there is a decrease in AMPK activation measured as a phosphorylation of AMPK during hypoxia exposures. And also, Dr. Burton published this paper where in humans, phosphorylated AMPK seems to be not changed, at least the ratio and also the phosphorylated forms. We did another study where we tested 3 different altitudes, so 100 meters in San Diego, Denver altitude, and Frisco. And we found that, and we include at least, these were higher sample sizes than the previous studies. And we saw that the ratio between phospho-AMPK and AMPK was increased even at a moderate altitude like Denver altitude, compared with sea level. So we also tested downstream pathways of AMPK, and we found different results. So one of the ones that that I would like to highlight here is TSC2. It's downstream of AMPK, and it's the connection between AMPK and mTOR. So we were very interested to see this effect. And there's an increase in TSC2 phosphorylation at high altitude compared with, and a moderate altitude compared with sea level. We also tested downstream of mTOR signaling, so for EVP1 and S6 kinase. And we saw that there was an increase in the phosphorylated form of for EVP1, which is a little contradictory with an increase in the TCS2, TSC2, because TCS2 should inhibit mTOR. So we think that maybe there is another mechanism to induce the activation of 4EVP1 phosphorylation. We then went back from the placenta to the maternal side, and we, as Dr. Julian showed before, we isolated peripheral mononuclear blood cells, and we analyzed the gene expression of different genes that are related to the MPK pathway. And you can see here that we saw an effect, a reduction, and these were at 20 and 34 weeks in these patients. And we saw a reduction in certain genes, like leptin receptor, that are related to MPK, so upstream genes. We didn't see an effect on the actual PRK1 gene. We saw a reduction in mTOR and also in X6. So when we, and this is also what Dr. Julian showed, we saw a correlation between uterine artery diameter in this woman and PRK1 expression. But this was true for high altitude at 20 weeks, but not at 34 weeks. And it was correlated with low altitude at 34 weeks, but not at 20 weeks. So as a summary, MPK sensitivity is increased in the myometral arteries in high altitude residents compared with the low altitude. And we think that this is through an activation of NO production. The placental MPK activation, it's an increase in high altitude compared with the low altitude. And in the high altitude residents, the maternal MPK gene expression is correlated with higher uterine artery diameter at 20 weeks, but not 34 weeks. And it's the inverse in the low altitude residents, where maternal MPK gene expression is correlated with higher uterine artery diameter at 34 weeks, but not at 20 weeks. And with that, I would like to thank all the people involved in these studies, especially Dr. Julian and Moore, that were the pioneers on all these studies, and I will take any question.